All right. Well, if everyone's here, we can go ahead and kick things off. I see the numbers are kind of creeping up, but I'll go ahead and get started. We are absolutely delighted to have with us today Professor Joel Freundlich, Professor of Pharmacology, Physiology, and Neuroscience, and also faculty member in medicine and member of the Center for Emerging and Re-emerging Pathogens and graduate program in medicinal chemistry. Joel is, of course, world renowned for his research. He had an undergraduate and master's degree in chemical engineering from Cornell and did his PhD work in chemistry at MIT. He's received a number of awards for his research and scholarship that range both from teaching awards, the Golden Apple Teaching Award in 2018 and 2020, and of course for his research. Most recently, he received the Rutgers University NJMS Basic Science Faculty of the Year Award in just 2023. He is an extremely prolific, highly active researcher with a well-funded research group that has an array of high-impact publications. And he'll talk to us today, I believe, about platforms in computation, chemistry, and biology for pursuits in chemical biology and drug discovery.
Great. Thank you very much, Darren. I really appreciate the kind introduction. Not sure what happened after by teaching after 2020 or so. No, I'm kidding. But yeah, it's always nice to be recognized by the students and your colleagues. And so what I'd like to do today is give everyone a sense of some of the work that we're doing. And please share. So I think I may have changed the title a little bit, but I think the idea is still the same, that we try to juggle different sort of technologies within the lab in order really, and I think stretching is a good word, right? It's tough to try to go from basic discovery all the way to potentially to therapeutics that impact the lives and health of people, but that's certainly those are amongst our goals. And so in thinking about the talk and sort of just the institute and quantitative biomedicine, if I'm remembering it correctly, I think we try to be as quantitative as possible. And so what I'll try to do today is talk about three sort of platforms in the lab and how we're using them in human disease research. One, to help better understand the biology of the disease state, but then two, trying to evolve therapeutics. And hopefully I'll give you a sense, you'll take away from this a little bit of our strategy and how we do try to be quantitative as much as possible. And obviously, right, the world needs new therapies, both molecule and target, and for many reasons, right? There's a lot of impetus for academics to be involved and for them to be active in this space. And thinking about this space, this paper, which now as the years are going by, we're on to I guess 19 years, still has had a really profound impact. So this was from former scientists at GSK, led by David Pompliano, where they basically looked at a set of gram-negative and gram-positive genes and essentially their gene products, where they had genetic data supporting, the importance. I'll be careful as far as essentiality or vulnerability. I believe it's more like in those days, essentiality, but really a prioritization of targets. And they screened hundreds of thousands of compounds per protein, most likely enzymatic assays. And the short of it is, despite that really significant effort, 7 years, multiple millions of dollars, they really had very little to show for this. Maybe five lead compounds. And so they were trying to be translational, trying to see if those targets could basically lend themselves to molecules that could have therapeutic potential. And so this was a very target-based approach. And the authors in this paper, in sort of discussing the outcome, really, came down on the negative side in terms of target-based drug discovery. And this paper has often been cited as one of the key papers that really pushed people at the time, and I think we're still feeling the effects today, towards really high throughput whole cell phenotypic screens, looking at what inhibits the growth of the bug in culture. So you're looking for a downstream property, growth inhibition of the cell, and you have the potential to find new drug targets. But obviously, there's lots of challenges here. There's a big time and cost, big issue with time and cost for screening. Your hit rates are typically very low, less than 1%. So you have a lot of inactives, and as you can imagine, in research, in most areas, right, of research, especially these days, are really, feeling the economic crunch. You're really only going to focus on those few actives. And so there's a lot of data loss. But nevertheless, there is this information, if captured, really puts you in that realm of big data, which obviously I think a lot of people at Rutgers are very interested in. And so it does provide a huge data set that can be leveraged, and we'll come to that in a minute. So we've really been thinking about this paper for a long time, and it's had a longstanding influence on the lab and the questions that we've tried to ask. One is, under what circumstances can target-based approaches still be viable, despite this, big data, high throughput whole cell phenotypic screening sort of push? Um, Secondly, we've tried to ask, well, okay, let's embrace big data, and let's ask, how do we best use that data to help find new active compounds and to evolve their properties in a drug discovery or chemical tool sort of fashion, or with those goals in mind? And then thirdly, more of a general question, regardless of platform, we feel like there's still a lot to be understood about how compounds accumulate in the cell type they're targeting, and if they undergo metabolism that can be linked to mechanism of action or mechanism of resistance. And so those are some of the key questions that have driven our research over the last probably decade. And so this has really kind of led us to try to really be helpful to the community, to churn out useful computational models, um, chemicals as tools, one, to probe biology, but then two, hoping that in some cases we're fortunate enough to find, our way to compounds with, at least preclinical promise, if not potential to actually get into clinical trials. And I'll tell you about one of those compounds today for tuberculosis. We focused on biological tools, such as those trying to measure drug accumulation within a targeted cell, and also try to discern metabolites. And then also, hopefully, our efforts have been, sufficient to help validate drug targets for us and the community to go after. And the real focus of the lab is infectious diseases. We've dabbled in cancer. It is certainly a long-term interest of ours, but the bulk of our work has been in the infectious disease area. And I think these numbers really, highlight that. Despite having gotten through, safely, most part, the SARS-CoV-2, COVID-19 pandemic, these numbers for TB and malaria, are, hugely significant, and these are basically repeating year after year. So TB is a pandemic. It's these more than 1 million deaths, more than 10 million cases have been going on for decades, and obviously we need to deal with these infectious diseases more effectively. And obviously there's a lot to be recognized as far as past efforts for TB. We have frontline drugs. And people may ask, well, wait, isn't this a solved problem? Aren't there drugs? And there are, but you have issues with resistance. So here I'm showing you 4 drugs, basically mechanism of action, but then associated closely mechanism of resistance. And in many cases, you're probably talking about hundreds of thousands of cases, per year worldwide for drug-resistant TB. This is, there's a need for new drugs to basically evade resistance, but then also to shorten therapy. We're talking at least 6 months of therapy. And, in these days where people can sometimes grumble about taking azithromycin for a few days, imagine being on TB drugs for 6 months. And then finally, I think we have to, in grants, make a push for why these diseases are of relevance to the U.S., besides us just trying to be good members of the global health community. We have outbreaks of TB in the US. I think this last year was, one of the largest outbreaks in history. So we're not immune to this, but obviously, we owe it to the world to help contribute to, a healthier, safer place to live. And so we'll focus on this first question to begin with. And as going through this, I tried to offer a new spin on this, and this is my spin, and I'm certainly open to comments from people. At the end, or if something's glaring, please, unmute yourself, and happy to chat about it. So trying to think, as we were looking at drug discovery and a target-based approach, there are multiple criteria that we try to juggle, and I think that how people do this is something that we hopefully all can learn from and think, and with the idea of evolving how we do this and hopefully doing this more efficiently. And so, for earlier stage, you obviously want to inhibit the growth of the bug in culture. Okay, in this case, I'm focusing on TB. So MTB is Mycobacterium tuberculosis. The MIC is growth inhibition. You want to do that without having a significant growth inhibitory effect on the mammalian cells. Vero cells are a model mammalian cell line. You want to understand target. And as a target, hopefully you'll come out of this part saying, yeah, target is really important. So you want to have this in 2 levels. One with the purified protein. If it has enzymatic activity, then you can get to things like a functional inhibition, IC50, or you can worry about a binding constant, for example, KD. Structural biology is hugely important. X-ray crystallography. So having a crystal structure of your target is hugely enabling. You also want to be able to see evidence for target engagement within the cell, and we'll come to some examples with this, but you can imagine drug-resistant mutants can be important, looking for MIC shifts. Um, in this case, we're going to be talking about a fatty acid biosynthetic pathway in TB, so you can actually do thin layer chromatography and look at actually what's happening inside the cell. So not only purify protein, but actually within the actual cell that you're targeting. And then I also threw as in sort of this fourth sort of square, in vitro ADME, so absorption, distribution, metabolism, excretion, kind of a big category. Physicochemical properties. Usually early on, what we would like to look at is mouse liver microsome stability. Since the mouse is our first efficacy model, you want to make sure that you can, with this phase, typically phase one oxidative metabolism, that your compound is stable. If not, in cells, with mammalian cells in the presence of your compound or in vivo, you're going to be getting metabolized. So you want to have some idea of that. And then also solubility is important. You don't want your compound to crash out and kill the animal or be very difficult to work with. As we move further into leads, preclinical candidates, etc., more criteria to basically juggle. Mouse PK is really important. That's our first efficacy animal. You want to be able to dose. Typically, we'll do oral. We want to get good exposure. You want to have your drugs stay around so that in vivo, you're actually have a chance for a therapeutic effect. That takes you to in vivo efficacy models. Obviously, you want to be able, that's really your first step in terms of trying to eventually get into a patient. And then we get into broader categories. Safety is very important. You want to avoid inhibiting or inducing CYPs. You don't want to have significant inhibition of hERG. You don't want to have an Ames positive compound or metabolite. And then you can get into broader panels that essentially look at common mammalian off targets. And then finally, in vivo safety is very important, right? And then you get into things in, your rodent species or eventually non-rodent, maximum tolerated dose, 7-day tox, 28-day tox, etc. So many things to juggle, often concurrently. And as you'll see as I go through the optimization, I'm going to try to basically yellow in or highlight the boxes that we're basically addressing at each stage, and hopefully that's is sort of interesting for people to see. So this is a cell wall program that's been a long-standing collaboration with David Aland, Matt Neidich, both at Rutgers, and then Matt Zimmerman and Veronique Dartois, now at Hackensack CDI. And then Eric Nurnberger from Hawkins joined the party a little bit later. And our lab was led by two former postdocs, Daigo, who's now at Schrodinger, and Divya, who's at Recursion. And essentially what was done in David Aland's lab was looking at this relatively small, maybe modest-sized number of whole cell active compounds against TB that GSK kindly turned out to the community in 2013, in most cases without any target information. David's lab basically profiled them using the INIBAC assay, which is a reporter-based assay that signals if the compound is inhibiting somewhere within cell wall biosynthesis. Okay, so it's kind of a broad phenotypic, kind of a pathway sort of based assay. This indazole sulfonamide, which was named by David DG167, came up as an active compound. We wanted to profile this, so we're starting to check the whole cell activity box, a good MIC for a hit below 1 micromolar, at least 10-fold higher CC50 against mammalian cells, so that's good. Physicochemical properties we're checking off, good solubility at least 1, if not 100 micromolar. And then we're looking at the mouse liver microsome stability, and this is poor. This is 10 minutes. This is below at least a half an hour, if not an hour. So we're being quantitative here in terms of metrics to identify an issue. By LC mass spec, we can see that this is due to oxidative metabolism of the 1-methyl group on the indazole. So that's a problem. Nevertheless, we wanted to see how far we could stretch with this compound. So we checked off the mouse PK box. I'm not showing you the data, but the area under the curve, this is 2 mice, a single 25 mg per kg dose oral, and then you follow the exposure over 5 hours. This is above 1,000 hour nanograms per mil, so this was not bad for a hit. Still limiting metabolism, but this is still modest for the PK. That allowed us with David Perlin and Steve Park to go into a subacute model of TB infection in mice. Here you infect, you let that go for 2 weeks, and then you dose once daily oral with compound. Here I'm showing you basically 2 weeks of dosing. The result at 4 weeks is the same, is that DG167 at 100 mg per kg once daily oral gave growth as compared to the bacterial load in the lungs, the CFUs, over time. So that's not good. You're not stopping infection. It's actually growing. Isoniazid as your sort of pinkish positive control is starting to give you sidality at 2 weeks, and there's sidality through 4 weeks. So this is a in vivo inactive compound. At the same time as we were getting our paper ready, GSK had published their results with the compound. Unbeknownst to us, they had a less conservative definition of activity, and they called the compound active. While we respectfully disagreed, we both agreed that the compound needed to be optimized. And so that's where the target part comes in. In order to optimize the compound, we felt that it was essential to understand the molecular target of the compound. And so essentially what Pradeep did in David's lab, he raised spontaneous resistant mutants in the H37RV background with a frequency of resistance of 1 times 10 to the minus 7th. He found mutations in this essential and vulnerable beta-keto acyl synthase Cas. It condenses an acyl ACP with a malonyl ACP to give a beta-keto acyl ACP that turns around in this fatty acid biosynthetic cycle, Fas2, to eventually give these long-chain fatty acids that are a key part of the mycolic acid cell wall. So we were very excited to know this was the target. This is starting now to check off this box as far as target within the cell. We have resistant mutants. There have been beautiful work with phthalactomycin inhibitors of Cas and plitenzomycin inhibitors of Cas, but unfortunately, none of them had sufficient properties to allow or enable an in vivo efficacy study with one of those compounds. So this is really an opportunity ripe for basically pharmacological validation of the target. And here again, we can see the resistant mutants where there's a specific, significant MIC shift for the compound as compared to the wild type. We further set up some tools for, again, looking at target within the cell. This is fatty acid TLCs. Essentially, you can subject TB to the compound. You essentially then lyse. You actually esterify the acids so they run up the plate. And you can see the mycolic acids. This is the end product of the pathway that we're inhibiting. And we see with isoniazid, which is a prototypical inhibitor, but inhibits a different enzyme than Cas, that basically the spot gets weaker. And so we're losing mycolic acids. The same thing with our compound. And then you see a buildup of these are shorter chain fatty acids. The spot gets darker with increasing concentration of drug. And so this precursor is building up as you're basically inhibiting its extension to the final product. So this is a nice way to basically tell within the cell that we are engaging the target. Excuse me, in this case, we're engaging the pathway. In this case, we use a spontaneous resistant mutant. Isoniazid has a different target, so it maintains this inhibition of mycolic acids, whereas now our compound does not have any inhibition because there's a resistant mutant inside the bacterium. So this is a nice check on target within the cell that we'll use throughout the program. We then turn to purified protein, and as I said, crystal structures. We love structural biology. It's so helpful, even though I just blew through the slide. Glenn Capadagli was a postdoc with Matt Nidich at Rutgers. He's now at Tentarix. And solve the crystal structure in the unit cell. There is a homodimer. We are binding in the acyl binding pocket. The malonyl binding pocket is down here. And interestingly enough, there are two molecules bound in the active site interacting with each other through an intermolecular hydrogen bond. So we now have a structure to work with. Just quickly, I'll talk about that when we didn't know the target of the compound and we didn't know the structure of the compound bound to the target, we were operating blindly. And so here when we're trying to basically improve the mouse liver microsome stability, because that was a key issue, and we're just checking in on MIC and we have no knowledge of target, basically, we were not able to improve the mouse liver microsome stability. You can see we made changes just on this methyl group that gets cleaved. We not only didn't improve mouse liver microsome stability, we also lost whole cell activity. So again, not knowing the target, not having the structure of the compound bound to the target, really sort of challenged us. Same thing, and then once we knew the target of the compound had a crystal structure, we can see the 1-methyl group pointed to this shallow hydrophobic pocket. So for sure, you're not going to be able to bulk that up because you're going to get steric clashes. So again, knowledge of this target and structure of the compound bound to the target, hugely enabling. And then the same thing on this other part of the molecule, the sulfonamide tail. We were just trying to look at whole cell activity without knowing the target of the compound and having the structure of the compound bound to the target. We could see some trends, which I won't go over for the sake of time, but we were really operating blindly. And Again, once we knew the target of the compound and had the structure of the compound bound, we could see that that butyl group points into a narrow hydrophobic channel. So it makes sense you're going to have a size limitation, and you're probably not going to be able to introduce a ring into this part of the molecule because there's just a lot of steric constraint around it. So hugely enabling. And really what this did was allow Daigo to come up with a design hypothesis. Here, he basically said we would transpose this nitrogen from where we have a labile, metabolically labeled nitrogen carbon bond to this position in the ring. So we could have either another indazole if X is a nitrogen, or if X is a carbon, we could have a series of indoles. And so in the lab, we do a lot of chemistry to make compounds. We aspire for our syntheses to be rather short and modular. The synthesis of the indole, which I won't go over at the sake of time, but if people have questions, I'm happy to go over that, is a little bit lengthier, but it allows us to install a nitrile on the 2 position, which we were interested in, and then have a range of sulfonamide groups. And then the indazole is very quick, and I'll cover this just because this is brief. Essentially, this chloroacetophenone can be condensed with hydrazine to give the indazole. We then use palladium on carbon to reduce the nitro group to the amine, and then we can react that with sulfonyl chloride to give the sulfonamide. So here again, knowledge of the target and paying attention to the mouse microsome stability, the metabolic stability is what drove these designs. And the short of it is that over about 200 compounds, we came to 3285. And in this case, we have a transposed nitrogen, so we have a much more, hopefully a more metabolically stable carbon-carbon bond. And now we have a 4-fluoro group on this butyl chain of the sulfonamide. And essentially here, we're paying attention to whole cell activity, and we can see that got better, 0.39 to 0.2. We're paying attention to solubility. That got better, 324 micromolar to 483 micromolar. The key property we're after, mouse over microsome stability in this box, we're almost at that half an hour, just almost there, 28 minutes. So that was an improvement. But we also checked in periodically on the PK. And now we can see we gained a 10-fold improvement in that area under the curve. This is a 0 to 24-hour exposure in mice. That, I believe, is really the main factor that's driven this compound to where it is today, is PK. So this box, I'd say, is hugely important. During this optimization, we really did not check in as much as I think we should have in retrospect with with purified protein engagement, either through a KD or an IC50. This compound in the end has a 71 nanomolar binding affinity. Its functional addition, this assay is a coupled assay. It's a bit of a bear. We only came up with this recently. We still need to titrate down further in terms of enzyme concentration. So right now we're sort of bottoming out at the enzyme concentration of 200 nanomolar. These in our second generation programs are trying to check in much more often. We did have crystal structures, and that was sort of way we were looking at engagement of purified protein. The nice thing is the crystal structure actually shows that it is as designed by Daigo. Because of unfavorable interactions, we only fit 1 molecule into each of the monomers, not 2 as within the early hit. But it's making those key interactions. And then checking in on engagement within the cell. We raised spontaneous resistant mutants with a frequency of resistance of 10 to the minus 8th. We saw mutations in Cas. These compounds were cross-resistant with the initial hit. And we get that same Fas2 phenotype as far as reduction in the mycolic acid and increase in the short-chain fatty acid precursor. And this goes away with a 3285 resistant mutant. So we were able to check in with the target engagement in the cell. So ultimately, paying attention to these properties got us to this compound. I'll just talk very briefly, but the compound looks quite promising as far as its in vivo efficacy. Now, if we go back into that subacute infection model, we see at 100 and 200 mg per kg once daily oral, we get cytality with 3285. We go, again, with David Perlin and Steve Park into a chronic infection model. Here you're infecting, letting that go for a month, and then you dose once daily oral for a month. We have a minimum effective dose giving you a 1 log reduction in colony-forming units in the lungs of 5 mg per kg. If we go to 20 mg per kg, we are equipotent with frontline drugs isoniazid or rifampicin at 10 mg per kg. And because TB must be treated in combination, we start to see some evidence that it can play nicely with, for example, isoniazid or rifampicin in very simple, just 2 drug combinations. So we've now basically have an in vivo active compound, and we've provided, I think, that much needed pharmacological validation for this target. So honestly, no matter what we do, I really don't care if we succeed or someone else succeeds. I feel like the path is there towards a Cas targeting drug for TB. And really the data that I think cements that is from Eric Nuremberger's lab at Hopkins, who joined the team a few years in. And so this is led by Hina in his lab, looking in that subacute infection model, now with more relevant combinations. Here we can see bedaquiline, protomidate, linezolid, which is a standard of care for multi-drug resistant, rifampicin resistant TB, but has issues with linezolid mitochondrial toxicity, and also questions about durability without having a fourth drug. So we can look at its BPA backbone and see that we can basically add 3285 at 30 mg per kg, now known as K. So BPA-K gives a significant improvement after 1 month of dosing in this model as far as the colony forming units in the lungs. And we can take the the actual used drug combination, BPL, and add 3285 to that to get BPA-KL. And now we have, again, a significant reduction in the TB burden in the mice after 1 month of treatment. And then we can also look at BPMZ, bedaquiline, protomidate, moxifloxacin, pyrazinamide, which looked quite good in the Simplicity TB trials, but there are issues as far as adverse events due to hepatotoxicity due to protomidate and pyrazinamide being used in combination. And then also in parts of the world, there's moxifloxacin monoresistance, which has to be considered. Here we can see that we can replace protomidate with 3285 to get BZKM and have a significant improvement in activity after 1 month of dosing. We can also look to an earlier stage combination, bedaquiline, protomidate, rifabutin, and add 3285 to that, and after 2 months of dosing, get a significant improvement in activity. So multiple regimens where 3285 can be added in or substituted in to give you improved efficacy. And then the icing on the cake as far as efficacy studies is in this gold standard mouse relapse model. Here, but basically dosing for 1.5, 2, or 2.5 months. You give the mice a 3-month drug holiday. I don't think they get to go to the beach or anything, but they get a 3-month drug holiday. And then you plate, and essentially a successful outcome is either 1 or 0 out of 15 mice relapsing. Okay. And so again, let's revisit bedaquiline, protomidate, linezolid, used for MDR rifampicin resistant TB. This is a 6-month therapy. In mice, after 2.5 months of treatment, you have 9 out of 15 mice relapsing. But now we go to adding 3285 to that to get BPLK, and we have a successful outcome, 1 out of 15 mice relapsing. So that was a successful outcome, as was, again, substituting in 3285 into the BPAMZ regimen, where at 2.5 months of treatment in this model, you have 0 mice out of 15 relapsing. So essentially 2 different drug combinations where adding in 3285 gives significant evidence for treatment shortening and real clinical utility. And again, the outcome is the same in the second mouse relapse model from Eric Nuremberger's lab. Here, he just did a dose titration of 3285K from 1 to 100 mg per kg, and we can see successful outcomes with 3 months of therapy at 10 or 30 mg per kg. So more to be done as far as dosing, but certainly this looks good that we can go down in dosing level of 3285 in this BPLK regimen. And so in this sort of short synopsis, hopefully I've emphasized to you the importance of multiple criteria in drug discovery, but then also really important to target, not only knowing the target of the compound, but having a structure of the compound bound to the target. Thanks to Matt Nidich and his lab, we probably have 10 crystal structures of compounds bound to Cas, and these have been hugely enabling. So I would give this a resounding yes, that target-based approaches still can be viable in this multiple criteria decision-making problem. And right now with 3285, we're currently in IND-enabling tox studies. We're hopeful we can go the distance. If we can't, we think that we've laid the groundwork for at least someone to come up with a Cas drug. Okay, so that was target-based drug discovery. Let's take the opposite sort of approach and say, yeah, we really like screening, and we're all in this age of big data in our lives at work and outside of work. How do we leverage that information? And so what I'd like to talk about is using machine learning models. We've been doing this for more than a decade. I'll show you the progression in our lab. But really what we're after is how do we take data that's out there and publicly available resources? Kemble, and I don't have the PDB on here because we don't use it in that regard, but that's obviously an immense, just treasure trove of structural information. And so it's out there, we just need to use it. And can we use information that's out there to find early compounds like DG167, but then also be able to optimize them. So it's finding the compound and optimizing it. And there's many people who, do machine learning. We don't pretend to be computer scientists. We're learning as we go along. And I think that's really a good aspect or a way to think about machine learning is we're improving on some task with experience. This is a nice quote from Kilian Weinberger at Cornell. And won't spend too much time on this, but basically, we'll use both regression models and binary classifiers. So we're thinking about a continuous variable that we're trying to predict, or essentially a yes/no. And the work has really sort of spanned in our lab, it's really been learning, going from prepackaged software like Discovery Studio Pipeline Pilot to now we code everything in Python. And even I am learning Python and getting better. Sometimes my lab may differ with me on my opinion. And then we're also going from very simple descriptors, ones that would, for example, just be used for docking, to much larger sets of descriptors such as in Mo or RDKit, and then also looking at more complex fingerprints or a wider range of fingerprints, such as those available in places like Mordred and other places. Standard sort of statistics to basically give us an idea how these models are performing, both binary classifiers and regressions. And then again, we're really after the application here. And so we've been able to find active compounds against a number of organisms and then understand something about their mechanism of action or mechanism of resistance. And I'll tell you quickly about the staff work. This was led by Jimmy Patel, who was an MD PhD student, who's now at Emory as a resident, and Alex Perriman, who was a postdoc, who's now at Variational AI. And we wanted to come up for obvious reasons, a model that learned about inhibiting drug-resistant Staph aureus. So we're basically grouping all, methicillin resistance, vancomycin resistance, linezolid resistance all together. And PubChem was a great source of information. 22,000 compounds got, winnowed down to about 1,600 compounds. And it was a fairly balanced data set. Actually, we had a lot more actives than I think we anticipated. More than half were actives. They built a Bayesian classifier with this and scored a 3 million compound diversity library from enamine. And here, what we were happy to see is that the top scoring compounds, okay, most likely to have activity, and here our activity was defined as an MIC less than or equal to 10 micrograms per mil. 3 out of those 15 had an MIC at or below 10 micrograms per mil. So we're not right all the time, and we never had that expectation. We see an enrichment in actives, but I think perhaps more importantly, the bottom 8 scoring compounds, the compounds least likely to be active to meet that 10 microgram per mil or less threshold, all 8 of the bottom 8 scoring were inactive. MIC at or above 50 micrograms per mil. So it kind of helps us focus on the compounds and takes 3 million compounds down to essentially, 15 and then 3. And I think that's really where we see and believe there's a benefit. And so the compound that we liked the most out of the hits was this quinoline, 3151. It had a single-digit microgram per mil MIC against MRSA, okay, so methicillin-resistant strain, as well as a methicillin-sensitive strain, and didn't inhibit the growth of vira cells. Well, how does it do against drug-resistant strains? These are a set of, or 2 sets of vancomycin-resistant strains, VRSA and Visa strains, vancomycin intermediate resistant strains from Barry Kreiser's lab at Hackensack CDI. And 3151 maintained its activity. So the model did what it was supposed to do. It found a compound with activity against drug-resistant staph that people were not aware of before. Still, we wanted more. And I think this is where we get to the multiple criteria of drug discovery. I'm not showing that sort of, figure, but we're trying to look at mouse liver microsome stability. Poor, 1 minute. Solubility, poor, less than 0.06 micromolar. We even stretched to PK, and this is poor. Not surprising since the compound's not very soluble and doesn't have great metabolic stability. We still tried to ask more. We went into a mouse MRSA peritonitis model with Steve Park and David Perlin. Here, vancomycin is your positive control at 100 mg per kg sub Q. 3 days post-infection, obviously your positive control works well. We see some activity, not great, but there's still some activity of our compound with a very, very much a suboptimal formulation. We can only get 24 mg per kg to IP as a formulation. So my point here is that despite all these shortcomings, the compound still showed some in vivo activity. And so we really think there's some value in this compound and in its mechanism of action that we're still trying to understand. And I want to highlight here that drug discovery involves multiple criteria. If the compound failed in all these other areas, but we didn't try to predict those. And I think that's a key aspect, and we'll come to that in a few minutes. Okay, so very much shortcomings, but well, I've shown you example where big data can be leveraged to use machine learning to find new active anti-infective compounds. But the compound had problems, no offense to the compound, and we need to evolve it. And so we come back to some of these criteria that I was talking about in the first part of the lecture, and we've built machine learning models and published them, and we try to use them in our own research. Mammalian cell cytotoxicity, metabolic stability, mouse over microsome stability, solubility, and even in vivo properties like mouse PK. And so, can we put them to use? And I think the answer is yes. We had this thionopyrimidine that, again, multiple criteria, and we can see the mouse over microsome stability, column is red. It's again poor. Turns out this ethyl ester gets hydrolyzed in mice, both in vitro and in vivo. Not surprising, with basically lysates from mice, different cell lines. Um, so we've tried to optimize this using medicinal chemistry, sort of rules of medicinal chemistry. And the short of it is that we came up with this 2-thiazolelyl methyl, and this was work by Xiao Gang, who was a former postdoc in the lab, who's now at Exelixis, sorry. And so this is a medicinal chemistry solution. We took a little bit in terms of hit in terms of activity. We're now at 16 micromolar MIC instead of 1 micromolar. But we now have a half-life above an hour, maybe a little bit ominous, 66.6, but nevertheless, this is a medchem solution to the problem. Alex wanted a computational solution. And so he looked at the synthesis and said, well, we don't know a target, so we have a lot of sort of flexibility here. And so let's worry about the ethyl ester that we know is labile. Let's come up with a virtual library of compounds. We looked at a small example from Sigma Aldrich of 411 compounds that hailed back through the chemistry to either alkyl halides or aldehydes and worked through the chemistry. And Alex said, well, if we could score this library of compounds with a machine learning model that knows about metabolic stability, and then maybe do some other filtering, we may be able to help solve this problem. Oh, Darren, did you have a question?
I was just noticing that there was a question in the chat.
Oh, of course.
Um,
Sure. Happy. Yeah.
so one of them was
Oh, sure.
the if you can read them, how many unique binding sites have been found in Cas A, and have any of these compounds? I missed the first comment, which was a couple minutes ago, but the
Okay.
second
Mhm.
question was, have any of these compounds ended up being a Payne's compound?
Good question. None of our compounds are Payne's compounds. As you'll see when I come to it, when we do broader based interrogation through machine learning models, we're always using tween 20 a la Brian Shoykit, so to make sure that we don't have aggregates that are promiscuously sort of inhibiting the enzyme's functional inhibition. Um, there are there are 3 known binding sites. And we basically have interrogated all of those. Other questions?
I think
Nope.
we're good.
Okay, awesome. Great. Thank you. No, I appreciate the questions. And so basically what Alex ran through this machine learning model to score the 411 compounds. Yeah, so get back to there we go. And essentially what we looked at is the top 13 scored compounds with essentially a Bayesian classifier model for basically mouse over microsome stability. 6 out of those top 13 scoring compounds had successful half-lives at or above an hour. We looked at the lowest scoring, so the least likely to basically be stable according to the model. And 4 out of those 5 basically did not have sufficient half-lives in the presence of mouse over microsome. And then we also chose compounds at random, and basically only 1 out of the 5 were sufficiently stable. So it shows the enrichment that the model gives us. And then honestly, from a medicinal chemistry point of view, these were non-obvious solutions to the problem. These benzoic acids would not be ones that we would have thought of. The pyrazoles, yes. Perhaps this arine, no. This arine, no. So it gave us some different solutions to the metabolic stability problem that we really would not have pursued. And I should mention this, the synthesis of these compounds was done by Tom Stratton, who was an undergraduate, who then went on to work for Phil Barron, and then is now in pharma. So we've been able to use these models to start to look at more and more of these properties, okay? And there's lots of properties to juggle. And I think that's a really challenging part. And I'd love to sort of discuss this with people after the talk if they're interested. But so what we tried to do in coming back to this multiple criteria decision-making problem was to try to find compounds that engage the protein, that had either reasonable IC50 or KD, had good PK because PK was really important in the success of 32D5, and then also good MIC. So we're starting to deal with some of these properties in this sort of this problem, but not all of them. And so just quickly, this is work that we just submitted. Well, actually, we submitted a while ago, but we're just in revision. And essentially, with Connor Coley's lab at MIT, we did high throughput docking of an enamine 2.1 million compound collection using MolPal. We did that against the acyl binding pocket of 3285. So that's the target-based part. And this, through some filtering, gave us 956 compounds from the 2.1 million in terms of their docking scores. So that's target engagement. And then Vidong, who was a master's student, who's now doing machine learning at PayPal, and Alex, who was an undergrad, who's now with Cliff Berry as a postbac, looked at two machine learning models to add into the workflow. One to help predict or try to predict an MIC at or below 12 micromolar, and then two, trying to predict a hit-like mouse snapshot PK at or above 1,000 hour nanograms per mil. So two different filters. They then took these compounds once they're scored with these machine learning models through some different voting approaches to wind up with 93 compounds that we purchased and assayed. So 2.1 million to 93. Shivangi was a postdoc. Sorry, she is. Sorry, I've wished her for a faculty job, but not yet. But she's a wonderful postdoc if people are interested. And so experimentally, 18 out of 93 had fulfilled the MIC criteria, which is a really good hit rate of 19%. As I said, whole cell phenotypic screening, your hit rates are typically 1%. Mouse PK, we were really astounded. 41% met that criteria. And this is something that we're not aware of any other published study of trying to predict PK and seeing these types of results. The intersection gave us 12 hits. And we're looking now at target engagement with Melanie, who was a postdoc, who's now at Northern Arizona University, and Reshma, who's a postdoc, a senior researcher in Matt Neitich's lab. Interestingly, here out of the 12 compounds, this is a functional inhibition assay in the presence of tweens. So we're basically removing the possibility of promiscuity through of inhibition through aggregation. Only 2 out of the 12, showed evidence of having an IC50 at or below 25 micromolar. So that brings up an issue, and we can talk about this more later. The MICs of these compounds are at or below 12 micromolar. So it gives significant evidence that Cas-A is not the primary target of these compounds. Your MIC, you inhibit the growth of the cells better than you do they inhibit the enzyme, purified protein, okay? And so this brings up questions for us that we'd love to discuss with people who know more than we do. Does docking guarantee target specificity? Does it tell you if you dock against that target, that's going to be that target in a cell is going to be the primary target? But then it also comes back to trying to deal with these properties. How do we predict these more than one at once? And this, again, I think is an open challenge that people are still actively investigating. But hopefully we've shown you that we've come up with different models that can help evolve properties and that computational solutions can be really complementary to medicinal chemistry ones. And obviously there are others in this group, in this area, Regina Barzilai, multiple pharma teams, Connor Coley, who we collaborate with, and certainly others. And then finally, I'm just going to take about 5 minutes because I really want to leave time for questions. We've spent a lot of time looking at how you can understand to what extent a compound accumulates within the cell and does it get metabolized. Because in TB, there are lots of examples of metabolism in prodrugs. Pyrazinamide gets hydrolyzed to give the acid. Very complex metabolism of isoniazid, essentially this small molecule to this adduct with NAD plus or NADH. And so this was really an open question. And the work was motivated by Shin, who was a postdoc in the lab. And really what I'll just talk about quickly is that we were interested in this compound that came from our labs from machine learning models. And what Shin could do was essentially take a metabolomics-based approach. So this is a liquid culture-based approach standing on the shoulders of giants such as Josh Rabinowitz, David Botstein. Q Ri took this solid this growth on filter approach to TB, and Veronique Dartois had more of a liquid culture approach. But basically, you take your bacterium, subject it to compound, you take aliquots at different time points, you basically work them up, centrifuge them, wash the cell pellet, You have to lyse that cell pellet by, in TB, you have to bead, and then you can essentially take that extract with cold acetonitrile methanol water and interrogate that by LC mass spec. And so just quickly, what I'll show you is that you can look at the kinetics of your compound that you incubated with the bacteria. You can look at it, the drug level accumulate or change over time. So this is 0 to 24 hours. This is a wild type strain in red, so we see a kinetic profile. And this is a mutant, and we can see actually in this case there's evidence that more of the compound grows in over time if we have a transposon and activating that gene. And that gene is actually responsible in part for metabolizing this compound to give this metabolite where the nitro group is replaced by hydrogen. And again, using this methodology, we can see time-dependent growth in the wild type strain of this metabolite inside bacteria. And then if we inactivate one of the enzymes that's involved in its metabolism, we don't see any of it forming. So it's really useful in terms of looking inside the cell. We're not worried about what the drug is doing to bacterial metabolite pools. We're actually asking what is the bug doing to the drug. And really what this allows us to do is to, and we published this recently in ACS Infectious Diseases, is a work by Amir, who's about to graduate in another month and go back to med school, and then with his graduate with his PhD and then go back for his MD, and Shivangi again, who's a postdoc in the lab. We can measure drug levels in E. coli, and we can see the effect of efflux. Okay, so moxifloxacin is more active when we go from the wild type strain to an efflux mutant, delta tol C. The MIC goes down. And what we can do is actually show you that the drug level goes up inside the cell. So really, it's another way, it's a more direct way of actually seeing the consequences of mutations on drug levels within the cell. We can use this to look at drug combinations. This is obviously very important for infectious diseases. This essentially is looking at a decatrol, basically not an infectious disease targeting drug that's FDA approved, that was discovered by Paul Blaney to have an effect as far as increasing the activity of novabiocin against E. coli. We looked at this against rifampicin, and there is synergy as judged by the checkerboard. And what we're doing here is looking at the level of rifampicin within E. coli. Rifampicin is kept at 1 micromolar, but now on the x-axis, we are increasing the amount of indecatrol, which does not have activity its own. And you can see that actually in red, the level of rifampicin increases as the level of indecatrol increases. And so actually the compound is making rifampicin more active by essentially permeabilizing the cell wall and actually allowing rifampicin to accumulate to a greater extent. So it's a way to actually understand how drug combinations work if one drug is modulating the accumulation of the other or the metabolism. And so I'll just end with a little bit of teaser. I said we're really interested in cancer cell work. We'd love to talk with people doing cancer cell work. We're interested in hypotheses that we could use, but basically we can do this in cancer cells. Amir teamed up with a former master's student, Anish, who's just finishing up DO school. We can see imatinib just as a model drug accumulating within this human colorectal cancer cell line in a dose-dependent fashion. And interestingly, Amir found that we could use just a commercial kit for isolating or separating nuclear fraction from cytosolic fraction. And now in a dose-dependent fashion, Amir can show you what are the drug levels inside the cells, specifically within the nucleus or the cytosol. And so we could actually look at localization phenomenon of accumulation of drugs within mammalian cells. So that's a bit of a sprint. So again, I'm happy to tell you more about that if you'd like, but we really feel like we have tools to measure drug levels within the cell that really excite us as far as our applications. And again, right, just trying to have some impact by what we try to turn out to the community as far as molecules, machine learning tools, and biological tools. And obviously, Paul Farmer's quote is, quite important as far as trying to have some impact, no matter how small. I'm just a mouthpiece. I've talked about the people who've done their work, wonderful collaborators, NIH for funding, and you for your attention. Happy to answer any questions. Thank you.
Thank you very much. I had noticed I missed one of the questions in the
Oh, sure.
Q&A from Fabrice Leclerc
Sure.
that I wanted to maybe read. In your compound optimizations, do you consider the measurement of K on, K off to enhance activity or selectivity?
Yeah, great question. We have not directly. I mean, I think those could come out. I think of the microscale thermophoresis binding assay, which is what we typically use for binding affinity. Although sometimes, Matt and I just also turn to a fluorescence-based assay. So no, we haven't yet to date. We certainly take a note in the TB area. People like Pete Tong have done just beautiful measurements of on and off rates for the targets they've looking at and talking about things like residence time. Um, it's definitely something that we should consider more in the future. So it's a great question. We probably do need to use that more.
All right, thank you. Looking through the chat, some other questions were what machine learning model was used to filter the undesired hit?
The undesired hit. Oh, right. Okay, right. So, in this workflow, we actually used a range of approaches. Vidong and Alex did. In the end, we found, perhaps not surprisingly, that random forest models did better than some of the deep learning models. We looked at some of the message passaging neural networks, earlier version of Chemprop 1 point whatever. Obviously given Connor Coley's involvement. Um, so it wasn't surprising that the PK data set, is smaller. It's all compounds proprietary to our lab. It's hard to find PK data out there in the literature, even places like Therapeutic Data Commons. So that data set's probably about 300, maybe 300 compounds. The MIC models, yeah, were bigger. Those were probably about we used two different data sets. One was about 100,000 compounds. The other was about 300,000 compounds. And still, even with that, we found the random forest models were as good or better than the deep learning models. I should always, because I think we all probably, we read Pat Walter's papers and blogs and everything. And so we did not, that was kind of just coming out when we were getting this paper together. So to have to admit, we didn't do a rigorous comparison of models to see which, with Pat's statistical comparison. So I definitely give you that sort of admission, and it's definitely something we will do in the future. But yeah, the random forest did just as well or better than the message passaging neural networks.
Another question. You did testing in mouse liver microsomes, which of course is necessary in a mouse model. Did you do any in vitro PK with human P450 enzymes or microsomes? Also, you look at many combinations. Do the drug combinations affect the PK?
Yeah, great questions. Let's see. So we did human liver microsome stability on more advanced compounds. Obviously, something we worry about, but your first model is the mouse, and so if you're not going to be if you're not going to be stable in the mouse, then you're kind of sunk. We also did mouse hepatocyte, human hepatocyte, dog, rat, etc. So we did get that data, and in general, those stabilities were greater than those in the presence of mouse liver microsomes. Um, let's see. We looked at for more advanced compounds like 3285, we looked at CYP inhibition, which was all I think greater than at least 5 or 10 micromolar. And then we also looked at CYP induction, which again was minimal. I think maybe one isoform there was maybe like a 1.2x induction compared to control, but so there was nothing that was sort of concerning. But yeah, those are really important things to look at. And then sorry, combinations. Right. And so yeah. So those gave us a sense that in general we weren't expecting any significant drug-drug interactions with 3285, being used in combination with other drugs.
Okay. Another question for the machine learning model to screen compounds with the improved MIC values, are the identified compounds novel or analogs of existing active ones?
Yeah, good question. Yeah, so those compounds were novel. We were not aware out of those 12 any compounds that were previously reported in the literature to have activity. I'm trying to remember, in terms of similarity search, how, I think that some of the compounds were reminiscent. I'm being a little bit qualitative with that of other of some chemotypes that had shown activity, but were not like it wasn't like you just added a methyl group or you made one small change and that was the known compound. So they were there was some that were closer, but not but definitely not just simple analogs. Yeah.
Given the role of expert taste in setting up filters and designing individual modifications, what are the paths for scaling this approach?
Yeah, that's a really good question. I know that, for example, I think Genentech has published, I think, I'm I can't remember the person's last name, but it's a Genentech paper in TB, I believe, where they use like 20 or 30 different models. So it's just this huge workflow of different properties. And I think that scaling up needs to be done carefully, and I'm certainly open to people's discussion here. Who may know probably much more than I do here. I feel like there's a danger in just throwing every single model in one workflow. That I think the challenges are that some properties run counter to each other, right? What may be good for target engagement may be very bad for sort of solubility or metabolic stability, and so I think it just demands trying to we try to do essentially two properties here, right? As far as the machine learning models, MIC and PK, and trying to understand that non-dominated surface, right? What do the trade-offs look like amongst molecules on that surface? And then here, obviously, you know, there's a big issue with basically the target engagement. So and I'd be curious, what you think, Darren, or others. Like, I don't think docking guarantees target specificity, like unless you do some sort of massive counter screening. But
Yeah,
I think that's a big issue. And then, honestly, we're in the midst of getting binding constants for all these compounds, and also just trying to pick 12 compounds at random from enamine, the 2.1 million, which is obviously, kind of like 12 out of 2 million, but just trying to show how this does compared to picking in random. But I've seen people put up docking hits that have, I'm pretty sure I'm talking about KDs here, not a functional inhibition, anywhere from, sub micromolar to 100 or 200 micromolar. And, and I'm not picking on Brian Shoykit because he can't be, I don't think you can pick on, but, so like, work from Brian Shoykit where, he has compounds that have like a few hundred micromolar KD. Or maybe it's a functional inhibition. I may be mixing it up. But so I'm not sure really where we fall in that. And so I think that leaves it open to mismatches in the sense that if we're by machine learning trying to have an MIC at or below 12 micromolar, we usually think that your engagement of the target is probably at least tenfold better than your inhibition of the growth of the cell. Yeah, because you have to get in, you have to find the target, et cetera, et cetera. Asking docking to find, single digit micromolar hits, that may be, in the docking they did here with MolPals, people may know. I think it's a QVINA sort of sort of back end with the active learning approach. I think that's asking a lot. So and honestly, when we got these results, and I think I talked with Mark Mirko about this, which is kind of humorous given his recent editorial in J Medchem. And I asked him, I'm like, well, should we have been less stringent? Should we have maybe made this, like MIC below 100 micromolar? And Mark's comment was, well, but then you're leaving yourself open to a lot of junk, that you may not have very poor whole cell activity, and that could just lead to lots of other issues, a la his editorial. So I don't know. I'm really it's definitely, I think, highlighted that there's a mismatch here in this workflow, and that's really
mhm,
why I wanted to talk about it. That this succeeded in some ways and failed in others, but and maybe this just means doing more precise docking, or I'm not sure. I'm not sure, like maybe Darren, not put you in a spot where others maybe is that asking a lot to find compounds that have like single digit micromolar binding affinities for a target just from a broad screen?
yeah, I think that's kind of pushing the limits there.
Yeah. So, so at least the way I look at this is it cuts down space. We cut down 2.1 million to 93 compounds, and so I view that, that there was an efficiency there, and I think we found some active compounds. We have more to do. And obviously with any of these hits, if we can get a crystal structure, which we haven't been able to yet, that will help us move the compounds forward. So hopefully this sort of narrowed down 2.1 million to some starting points for more target-based optimizations. And that may be as good as we can do. But again, I'm always open to seeing how we can push these computational approaches, especially in concert to start to deal with these multiple criteria.
Mhm. Fabrice Leclerc points out that poor selectivity contributes to approximately 60% of drug candidates' failure due to toxicity and side effects.
Hmm.
And just to sort of finish up, we had two additional questions. One of
Hmm.
was how did you do the DMPK studies? Do you do them in-house or through a university core facility or CRO?
Right. So we've collaborated with Matt Zimmerman for probably the last 12 hours. Good. I'm thinking of PK being 0 to 24 hours. Last 12 years. So he and Veronique Artois were at Rutgers, and then they moved to Hackensack maybe about 6 years ago. So we've
Got it.
worked with them, and yeah. Yeah, and the one comment, yeah, and I completely agree as far as off targets. And so obviously having, some sort of filter from mammalian cell cytotoxicity, and typically we use Vera cells, African green monkey kidney cells. Um, so I think that can help you. But it's a really good point that's being made in that comment in the chat, and I think it points out to the need to try to do in vivo studies as soon as possible, because you never know, and you may just have something that just rears its ugly head as soon as you start dosing it in an animal model.
Got it. Last question is for machine learning models to predict microsomal stability, can the model suggest modifications or substitutions instead of predicting the stability of the submitted compounds by users?
Good question. Right. So we haven't, so right. So the way that we've run that model is essentially by um, run that approach, that workflow is by creating a model and then essentially then using it to score a virtual library. So we basically have done that work. I mean, certainly you can, so we haven't really gotten into generative approaches yet. We've certainly read about them, so I'm very interested in looking at that. And obviously, like people like Connor Coley, when he visited a few years ago, talked about generative approaches. So yeah, I think it's a really interesting area, and could we get to the point where you could use a large language model to give it a compound and say, find me something more stable, so I think it's moving that direction. But obviously lots of things to consider there in terms of synthetic feasibility, the actual target, the molecular target of the compound. So, but I think that's definitely true. I think will be a very fruitful area in the future.
Fantastic. Well, on behalf of all of us, we'd like to thank you for this very stimulating and lively talk and for your time. And so thank you very much.
Now, thank you so much, Darren. I really appreciate it. I appreciate everyone. If anyone has any questions or anything further, just please email me. And again, I'm hopeful I'll be down on the New Brunswick campus soon.
Fantastic.
All right.
Thank you.